Welcome to the actual von der Waal family suite. It's really nice to see real people in person. We're very excited to have you all here. My name is Rhonda Rosen. I am the Programming and Exhibitions Librarian here in Hannon Library. On behalf of our entire outreach team, I just want to say welcome, welcome. So tonight we have a very special program with Professors Demian Willette and Tatiana Kuzmenko, who will be discussing their publication Plant-derived Drug Discovery in an Introductory Biology Laboratory course. And they will show us how they developed a hands-on experimental laboratory module that introduces students to one of the many important aspects that plants play in our lives, plant-derived medicine. So a little housekeeping before we start. Again, if any of you are on honors and you need to sign up, right over here is just a little sign-up sheet. We want to make sure you get credit. We call this faculty pub night. Unfortunately, we have no pub, meaning, yeah, sorry. Well, we didn't want to mix it with the plants, and then who knew what would happen, you know, but it is pub night, and it is about faculty and our wonderful, talented faculty that we have here, but it's not just for faculty, so we're so glad to see so many students here tonight. On your chair, you may have noticed that there was a small little white feedback note. We would love for you to fill it out because it will help us plan programming like this for future. But also, if you include your email address on it, you are entered into a raffle and you could win a $100 Amazon gift card. So after the presentation, we will have a chance for you to ask questions. And after that, we will turn you loose for the wonderful specimens that our professors have brought with them. And we are recording tonight's presentation. And last but not least, I just want to let you guys know that if you have ever come out to Wellness Wednesdays out on the quad, you know the library has a table out there. And tomorrow for Earth Day, we will be offering free little succulents. So please come and get them while they last. Now, I have a hard time killing those. So we always like to begin our evenings with a land acknowledgement. So I would like to say that it is important to note that we welcome our guests onto land that is not ours. We sit tonight on the unceded lands of the Gabrielino Tongva peoples, who in the face of ongoing settler colonialism continue to claim their place and act as stewards of their ancestral lands as they have for the past 8,000 years. These lands reach from the Pacific Ocean, where we stand today, well into what is San Bernardino County. The whole of the Los Angeles Basin is unceded territory. I would ask you all to take a minute and spend some time thinking about what it would mean to take responsibility for one's presence on stolen land. I also encourage you to learn more about the history of the land where you sit by going to native-land.ca. So tonight, Professor Demian Willette is an assistant professor in the biology department at LMU. His teaching and research are in applied ecology, the field of taking science from the bench top to the real world to solve ecological challenges. Dr. Willette has his bachelor's and master's in biology from Iowa State University and Cal State LA, respectively, and his PhD in environmental health science from UCLA. He also did 2 postdocs in genetics and fisheries in Southeast Asia. He has taught and conducted research in 13 countries and has published over 40 peer-reviewed scientific papers. Wow. He is a Fulbright scholar, and his research has been featured by the LA Times, BBC, CNN, Fox News, NPR, among others. Tatiana Kuzmenko earned her BS and MS in evolutional biology from Moscow State University in Russia while studying social behavior of animals. Then Tatiana decided to dive deeper into the brain and got her MS in molecular biology from the University of Southern California. Tatiana enjoys learning and teaching about neurobiology, and she implements its concepts into her teaching style, engaging students through excitement and curiosity, the true drivers of learning. Over the past 9 years, she has been working with a great team of passionate LMU faculty and TAs on developing research-based general biology labs at LMU. Tonight, she's going to share her favorite research project, plant-based drug discovery in the Introductory Biology Lab. So, I welcome you both. Tatiana first. Okay.
Should I use a microphone?
No, that's John.
John? Approved?
Some of us are here even care.
Just for John. Good evening, everybody. I'm so excited to present something we really worked on hard for the last 4 years, and it came to fruit. And so many people here being part of this project, so I would like just to start with a little outline. First of all, I'd like to provide you some overview of the project, what it is about. Then I'll cover some of the lab methods that we use in labs. Then I'll show you the video tour of the lab so you can have a little peek into how it looks like during the labs, and you might see yourself if you're a freshman here. Then I'll talk a little bit about overlap between Chumash traditional uses of plants, scientific discoveries, and our data that students found in 111. Then Dr. Demian Willette will take stage to talk about value of active learning and applied science in teaching labs. And then you will hear some students' perspective. We have some TAs and students in the room who I hope can share what they think about plants. The whole story started with this publication. This is our alumni, Ashwarya. If she's not here, I'll definitely let her know that her face was here. And she was a student when we started working on this paper, and now she's alumni, and she's not around, but she was so much fun to work with. And we published this paper based on the project. I'm going to go over. So the general biology lab consists of around 250 students that are taught by 15 TAs and 3 to 4 instructors. Students work in groups of 3 or 4. Each group gets a different plant to study. Students design their unique experiment targeted to a plant they want to learn about. And most plants are grown in the garden, the one which right here. By now, we tested more than 50 different plant species, and some of them are here in the room. I hope you will take a look and smell of them. And I'm going to show you some of the highlights of the 4 years of data collections we did so far. This is our first hero, and I think the favorite animal of our TAs, Daphnia. Daphnia is a small crustacean. It will be right there on that demo station. It's barely visible by eye, but if you put it under the microscope, this is how it looks like. And you can see the heartbeat right on the back. And the little babies, as Roland pointed out. Three babies right there. So this is our hero that we test stimulant or sedative properties of plants. If the properties are sedative, the heart rate will slow down. And if the property is stimulant, like in coffee, the heart rate will go up. And we use methanol as a control, because most of our extracts are made with methanol. Our second hero is a brine shrimp. Brine shrimps are also crustaceans and almost transparent. They're very easy to study, and they are on the second table. You can take a look under the stereoscope. Students look at the toxicity properties of the plant using those animals. Their skin is very thin and transparent, and if they encounter some toxic compounds, they will slow down and die. So when we add plant extracts into the salt water, students can measure within an hour how many of them are still moving and alive. And based on those numbers, they calculate the toxicity of the extract. This toxicity was linked to anti-cancer properties of the plant. The last assay I'm going to cover is antimicrobial disc diffusion assay. This is the agar plates, and they are set up on the last table on the back. You will see for yourself. We used Bactine, a normal antiseptic, as a control here, and methanol, again, as a negative control. We're trying to make sure that methanol doesn't have an effect on bacteria, only plant components do. And you will see some of the antimicrobial activity on the tables behind you. So those are 3 assays. How they've been performed? Here we go. This is the garden. I hope you come check it out. It's located on the 2nd floor of LSB. Just, I suppose, do the scavenger hunt and try to figure it out. Or if you lost, go to room 216, it's my office, and I'll give you a tour. And help you name all the plants. This summer, we have a project to make a name tags for all the plants and the little properties of each one of them. So after the summer, hopefully you can go and see all the plants and description of them. And here we go. Most of the people here are actually TAs because I had time to take images of them while they practicing doing what they will teach their students to do later. So they harvested the plants and dry them, and then they grind them in a regular coffee grinder. And this is how mugwort looks like. Afterwards, they are stored. Yes. After you got the powder, you add some methanol, 100% methanol. That's what we use to extract the compounds from the plant. And afterwards, they mix and vortex it, and incubate, and mix again. And this is Camilla. Yes. This is how extracts looks like after the incubation. The blue one is not an extract. It's a balance for the centrifuge. This is part of the instructional videos that we make for our students, so they actually don't break the equipment they use. This is made from by Carolyn, one of the stockroom workers. She recorded this video. She shows how to extract properly. This is probably one of our TAs. Getting the extracts without disturbing the powder. This is the grind trim. And this is our growth room. This is how the anti-cancer assay looks like. This is Belan. She's not here today. So she's dispensing exactly 10 brine shrimp on the Petri dish, and you will see the Petri dish on the back afterwards. And this is our Daphnia. Some of the students, maybe here, maybe not. We evaporating the methanol, making sure there's no methanol left. And this is our Daphnia under the microscope. Students count the heartbeat, and it's very fast. It's actually a tricky thing to do. Here is a close-up of the heart. So you can see the animal is transparent, so you can visualize the heart very easily. And this is Suraj and Rukban. They're setting up the antimicrobial assay. They apply the extracts onto the paper discs and put them on top of the bacteria to be grown. Afterwards, we put them in an incubator, and after a week, you can see something like this. This is a new assay. It's yeast, and the balloons are catching the CO2 to measure the activity of the yeast in a bottle. Still to be worked out. It's a very new assay. We just invented it this summer, this fall. Still lots of work to do. But what did we find? Out of 50 species of plants, I highlighted some of my favorites here. And I sorted them by the scientific name. Why so? So you could see that by the scientific name, those plants actually got sorted into nice, almost perfect two groups. One of the Artemisia species and one of the Salvia species. Very interesting. We could do phylogeny of those plants to find out that they are related for some reason. So those which are highlighted with the stars, red stars, are present today, and I put the fresh plants on this table with labels. So please take, come, touch, smell, and learn more about those plants. But I'm going to talk about mugwort, white sage, black sage, and California sagebrush, maybe. So you could see what we found about them. So those are native Artemisia and Salvia plants that are found in California and been used by local Native Americans for thousands of years. Now they're in the lab, helping us discover new medicine. We've been using this book as a guide to discover native medicinal plants. I brought this book, it's on the second table. It's been written by James Adams, who used to work in pharmacology department of USC. He's retired right now. And Cecilia Garcia, she's a Chumash healer. So they got together and wrote a book about how tradition overlaps with biochemistry. Based on this book, we picked some of the plants that natives used and tested them in the lab. Which plants did we check? Well, one of them is black sage. Black sage was traditionally used to treat chronic pain, and the way how they did it, they made sun tea. Sun tea is made with ocean water and black sage left on the sun for the day. In the evening, people will soak their feet in this tea, and it will alleviate chronic pain. Additionally, it was used as antimicrobial for colics, cough, and sore throats. Let's take a look what science talks about it. Well, there is some evidence that black sage actually works in a number of pain receptors, including skin pain receptors and anti-inflammatory mechanisms. So it works through the skin into the brain and back into the back, treating the chronic pain. What did we see? You will find out just in a bit. This is the white sage. White sage is a close relatives, but they look very different. White sage used to be used for acute pain, especially some burns and skin-related pain. Additionally, it was as antimicrobial for sore throat, coughs, colds, and infections. Native Americans use it as a shampoo and deodorant, and they burn it in ceremonial practices. The simplest recipe I found in the book is related to white sage. You take just one leaf of white sage and put in your water bottle, and you drink this water throughout the day. And the next day, you just put a new leaf. One leaf a day. It somehow releases a certain amount of chemicals into your water, but not too much to make you sick, but exactly amount that Native Americans use it to disinfect their water and just keep it as a tonic. What's known about white sage? Here is an article I found, and it shows that white sage is known to have antimicrobial and cytotoxic effects. Cytotoxics, basically anti-cancer. Additionally, it has a very interesting influence on GABA, opioid, and cannabinoid receptors. This is very interesting because this is what affects our mood and have a sedative effect on the body. And the cannabinoid system is spread out on the whole body of this person, so you can see that this plant can affect many organs in the body. And this is what cannabinoids actually target, the endocannabinoid system. So what we found on those sages? First of all, both white and black sage have antimicrobial properties, and those are the pictures from the presentations of students from last semester. Additionally, they have sedative properties. Daphne heart rate slow down. And white sage also shows some anti-cancer properties. So it correlates pretty well with the scientific findings and the way how Chumash people used it. Another plant I want to talk about is California mugwort. It's my personal favorite. It was traditionally used as an antimicrobial and pain relief for toothache, gum disease, colds, compresses, for arthritis, as well as a very important plant for women's health and the menopause flushes. In fact, James Adams, when he came here to give a speech, he told us a story that every night he makes his wife a mugwort tea, putting one leaf into her cup. She says it smells and tastes disgusting, but it works. So she's been taking it for a while, and that's what helps her to deal with the menopause. Additionally, mugwort is known to promote very good dreams, and people make pillows out of it to get some lucid dreams. And Native Americans chewed it to help deal with some addiction like smoking. The science on mugwort is also very, very broad. Here I found an article that talks about cytotoxic effects of mugwort. This is the antimicrobial effects of mugwort, and here we go, some psychological effects of mugwort on ADHD. What we found? Well, we found all of them. We found that mugwort has antimicrobial, anti-cancer, and sedative properties. I have examples of student presentations here with the credits on below. This one is regarding the heart rate on Daphne. It has a sedative effect, stronger effect if you use essential oil of this plant because it's more concentrated. And this one covers the brine shrimp toxicity. And you could see that with the time brine shrimp dies if 100 microliters of this extract is applied into the dish. You might be wondering why some plants have such a dramatic impact on human physiology. Do you wonder?
Yeah.
I do. As a biologist, evolutionary biologist by training, I was actually very curious why plants have such a huge impact on us. And one of the working hypotheses, which makes sense to me, is a herbivory management hypothesis, which basically says that plants evolved with herbivores for millions of years, and they had to adapt to each other. They had to regulate their behavior in order not to be eaten too much. So they developed so many mechanisms. That's why it's so complicated when we try to break it down. The chemistry is so complicated to regulate the behavior of herbivores so they don't eat them too much. And this mechanism goes so deep into our physiology that even though we don't interact with those plants on a daily basis, our physiology is still using the same mechanism as herbivores do, including insects who also eat plants. So that's why Daphne heart rate still responds the same way as a human heart rate. The mechanism is still very similar. One of the examples here is a theogen. It's a ketone found in Artemisia and Salvia species, and it's toxic if it's consumed in large amounts. So it's a medicine, but if you eat it too much, it's toxic. It's perfect to control the herbivore behavior. They know that they ate too much, they had a stomach ache, they stopped eating the plant. This is how plants regulate how much to be eaten to. But at the same time, animals learn how to use those plants to treat their own disease. It is known that, for example, horses will eat wormwood when they get intestine parasites. To get rid of the parasites, they will eat certain plants. And the reason those plants smell so well, and I hope you all will smell them, is because they have a very complicated communication mechanism as animals. So all the smell is meant to let them know what kind of plant they're eating and what kind of chemicals are inside this plant. So there is a lot of evolution going on behind it, and I was very excited to learn more about this. And as you may know, I didn't say anything about teaching yet. And this is a paper about teaching, because I saved it to Dr. Willette. But to summarize my part, I think that we are here presenting the great screening tool for new medicinal plants, because we see that tools that we use in the lab correlate very well with scientific results and traditional usage of plants. Additionally, it's a unique educational tool that could be used in the general biology settings. And what's next? Well, we want to retest the most interesting plants and separate specific compounds. And I know I have at least 3 students here who is going to do 4 students here. Oh my God, all of them here. Oh my God. You guys, can you stand up so people know who is going to do medicinal plants in the summer? Yeah! Okay, you can ask all sorts of questions to those. They will be doing all the assays that I talked about in the summer, confirming the properties of the medicinal plants. And I would like to give some control to Dr. Willette.
Thank you, Tanya. No pressure on the 4 of you to make sure that all the results come exactly like how they did in the past years. No pressure, no pressure. And so it's a beautiful introduction or discussion of what we try to accomplish, but it actually has this backstory of this class which we teach, which is the first biology lab course all of our biology majors take when they enter into LMU, as well as you saw there were 250 students in that class. That goes well beyond just our first year biology majors, but also a number of sophomores, juniors, and seniors, as well as many other departments across the campus. And I'll give you some of those numbers later on. This actually started years ago, was when I got to LMU 6 years ago. We had this class, Tanya and I were working on it. Dr. Lum was also part of it. And we kept struggling with the plant component, because there was a question, how do we engage students with plants? The bacteria is really interesting. We had some assays that we could do there. The animals, of course, they're very interesting, but we struggled with the plants. So what was interesting is that this work was actually the 3rd iteration of how we attempted to solve this problem of how do we engage students about getting them excited about plants. And so this was one of our takes, and this is the one that stuck, and hence why we kept repeating it over 4 years' time to improve it. And so the class is this Bio 111. This is our General Biology Laboratory 1 course, okay? And we really tried to focus with this idea of inquiry-based approach, particularly because it matches what our goal was in the course, is how do we introduce scientific method and some of these concepts that I'll mention later on. What's nice about this is that in some sense, we're able to shed the responsibility of the education of what students pick up from us having to lecture to it, because it's a lab course, and putting it on them to drive that. And so that increases the responsibility of the activity, both in the design and scope. So they're given tremendous freedom, which has pros and cons. I'll get to there. But we kind of drive around these 2 ideas, is that we start by this idea of structured inquiry, that we're going to prescribe some things to walk through, to give them some scaffolding framework, and then we let them loose. And then we let them loose on the second part of it. And we found that's actually worked really, really effective, in part because we have TAs who've actually taken the course, gone through some of those pains, and then they're teaching what they discovered. So now that we've done it 4 years, we have this institutional knowledge that gets cast along without them giving the answers. We're not hand-holding them through it, but instead carefully guiding them through the process. And that's where I think we get more out of it. So we get these benefits, this idea of shifting the emphasis to learning and discovery, and this idea of encouraging ownership of the experience. So we can see some of the benefits. There's lots, but a couple listed here is this shifting on this process of learning and discovery rather than getting to some result. We actually don't know the result. That's why we've studied 50 plants, because every time we bring a new plant in, we don't know what it's going to give us. We don't know if we're going to match the literature or be something new. And again, that ownership of the lab experience, the model that this is set up is that we actually have 3 identical laboratories set up, each with about 16 students in them. There's one faculty that rotates between those 3 rooms. There's no way that faculty can be in every room, so we rely on our TAs. And they rely on each group to drive this. These are great. It's great for any lab experience. This is what we're trying to do, is this genuine lab experience. But it also helps with their independent research that they encounter later on. So this is one of my students, Sydney Lee. She graduated in 2020, and what you see are these tanks. we kept killing all the Daphnia. We could not figure out how to culture them. She comes with an Aquarius background, and she solved some of this. And this became one of her research experiments that she set up. A lot of the students who take this class and go on to be TAs and researchers, they do their own independent research, and just last week, they were at the West Coast Conference for Biology, and the LMU students swept across many categories in winning in terms of presenting their research amongst many other universities in the United States. They carry these ideas and then develop the research. So we think it's not us, it's their advisors, but we like to, get them started in this direction. Now, what are the downsides of this? Well, include some initial cautions in terms of how much information we give the students. We do this survey in the first week of the course where we say, we ask questions, and most students rank really high when we say, I like to learn things on my own. But then the other thing that later in the survey that they say that gets ranked equally as high is, I like to be told exactly what I need to do to get a good grade. And those two things are an opposite. And so it gets this idea of how much information do we give them, how much do we guide them, and then this idea of the second point, this occasional frustration or discomfort by not having a correct answer. We don't say it. They're like, well, why is this working, or what should I discover? We don't know. We'll get you there through this process. And this general idea of real science, which is messy. From here, we think plants are awesome. Tanya and I do. But not everybody thinks so. So plants can really be this challenge throughout education in biology instruction. And so we look to increase that to the level of how students are excited about animals and so forth. So plant studies play this really essential role where we see in some biological concepts, such as genetics with Mendel there with his genetics, but most biology teachers will defer to animals to give examples. And that's tricky. George Washington Carver is one of my favorite because the first STEM class I ever took, the building was named after him. And he did all these things with plants, which I didn't like plants until much later on. And it's like, oh, that's the building I was in. And he was an alum of the same school. He went to Iowa State. That's where he got his degree. And became the first, one of the first professors to teach about the plants in that way. And we get to this idea of plant blindness, that there's all these things in biology, but if I ask you to talk about some things in biology, you'll probably skip over the plants. You'll talk about the good examples that you know of animals. And so how do we adjust that? How do we get this idea where we increase their interest in plants? Well, we can tie it with this idea of medicinal plants and herbal drugs to kind of get that as a foot in the door about plants and then to open up new opportunities. And so I mentioned this idea of this inherent challenges of doing science to understand the unknown. And that's where we drive this hands-on inquiry-based strategy, which we find very effective for first-year students. We realize our students come from all over the world, all over the United States, with a varying level of introduction to biology. And you can see that over here. Students who have taken our class over these 4 years come from 36 different departments on campus. They're not all biology. Most, 31% are, but you can see a lot of health and human sciences, and then throughout the university. I'm just saying that they're not all freshmen. Most are. But we also have folks who already have a bachelor's degree and come back. So this needs to be widely applicable, and so an opportunity to learn from them. Again, we do this idea of leveraging a topic that's exciting or familiar, medicine. Walk in the door, and 80% of our first-year biology majors are medicine. When they walk out the door, that's not where they're going. They've discovered many more things that they prefer to do. Using that to dive deeper into plant biology. And that gets to our goal as a biology department, this idea of running towards this idea of teaching this experimental design, scientific method, and these ideas to appreciate the fact that doing science is messy, and you don't need to know the answer to get started. Okay, and so that's all I have on education. And I'm going to hand it back to Tanya because
we have some TAs in the room and students. Yes, all 4 of them actually present. I didn't know who is coming, but apparently all 4 of those are here. And what we're going to do right now is first of all, I'll ask all the TAs to stand up so you know who are TAs. Then I'll ask them to share just a little bit of their experience in front of the group. And then we will spread out and have questions and discussions around the demo stations, and you can approach those guys and ask them all you want about plants, and hopefully they can respond. So let's do this. All the TAs for Gen Bio, could you stand up and just say hi? And in the front, look, they're just right here. You can see the faces matches. Those are winners of our LSB Olympics. And good freshmen who took general biology recently, stand up. And dun dun dun, look how many are here. So you can ask them about how was their experience with taking the lab. And let's see, who from the TAs would like to speak? Maybe multiple? 1, 2, Camilla's looking at me. Okay, Camilla, come over.
Hello, my name is Camilla. I'm a senior. I've actually been a TA for 3 years, so lots of experience both online and in person teaching this. And it's been really fun. There's challenges, but once you get to see your students really engage in the scientific method and get excited about research and not knowing the answer, it's really, really cool to see like their minds just grow as scientists. So that's probably the best part about being a TA.
Okay, anybody else wants to? I think Veronica wants to say this.
Yeah.
Go.
Hi, I'm Veronica. I'm a senior. I'm a TA. I've been a TA for like 8 months. But I've been on the teaching track since I was a freshman, so I've known that I've wanted to teach since I was 18 years old. And my background is with 4 and 5-year-olds, so I came into freshman lab and I was like, yes, 4 and 5-year-olds. That was not the case, although sometimes they do resemble it. But I had a really fun time showing the students from an education standpoint that not everything works out the way that you want it to, and it's this is science. This is what you learn, and I think what's hardest for them to conceptualize is that even though their data may not be, they always say, my data is not significant. And it's not that your data isn't significant, it's that it's not significantly different from the control. And I preach that to them all the time because, yeah, your data is not different, but that doesn't mean anything. That means that you just discovered that maybe that isn't a cure for something, maybe that isn't related to cancer, or maybe that isn't related to an antimicrobial property or whatever. But I think it's really interesting to actually find the data that does not, or is not significantly different from the stuff that you're actually looking for. So yeah.
Well, tea is good. I was going to ask one of the students to share their experience taking a class. Let me see, I'm looking at Kristen.
I think Assad wants to over there.
Well, any student who wants to share their experience very quickly, how you encountered plants, medicinal plants. Maybe? Okay, go.
Hi everybody, I'm Saad. I'm a freshman taking Gen Bio Lab 2 now. I took Gen Bio Lab 1 last semester, and I wasn't really interested in plants, and I kind of had to power through it, but by the end it did start to get more interesting. And it is a process, and it just takes time, and you just have to appreciate the journey, not the end point.
Anybody else? You're good? Kristen? Well, Kristen took the lab online. That's why partially I wanted her to share her experience, because doing biology labs online is very difficult. You have to set up your own experiments at home, from scratch.
So I did bio online, and I chose rosemary as my plant, and I was it was a group project, but I ended up doing the majority of it. But so we grew yogurt in my kitchen, which my mom hated because it smelled really bad. But I did find that I had interesting results with the rosemary, and that things that I expected did not show up, which may have been due to my kitchen, but also that got me interested in doing syrup this summer, because I found it interesting, the stuff that I saw, and I wanted to see if that's confirmed in an actual lab. So yeah, that's what I'm doing syrup.
Thank you, Kristen. There you go. You can do experiments in your kitchen. Oh, you want to talk about yoga?
Yeah, so you bring up a really good point. Is so when we went into the pandemic, we had to shift and take this model online. And how do you do that? Well, you borrow students' kitchens. And so one of the things is we took this idea and we leveraged one of the faculty who was able to, whose dad was really good at fermenting things, including making yogurt. So we had him make a video of how to make yogurt, and then we developed a whole bunch of assays students could do with things around their kitchen, the classic, kitchen sort of science. And we still did this idea where they were able to get plant extracts. We taught them how to do that at home. And then they did the experiments where they grew the yogurt, and the yogurt is driven by the bacteria. So if you add something that inhibits bacterial growth, you just have a bunch of milk, as opposed to the yogurt fermenting, and that's the smell that folks talked about. It didn't work out the way, and that's actually a success. So you smell, if it smells bad, you're like, hooray, that worked. It's got antimicrobials. And then you saw the yeast example with the balloons. That was another one that came from there. And so that was a spin-off, is that how do you then take something which is in person and do it virtually? And I'll say that next month that paper is being published as well, because we were able to leverage what the students did and so forth. So we'll share that as well, same journal, but the idea that these aren't isolated to the lab, we can take it into different media and do it virtually, and the concepts still carry over. So I do want to share that it moves around in terms of how we apply it. So we did want to ask, do you have any questions from the audience for Tanya? Or?
Well, we have acknowledgement.
Oh, sorry, acknowledgement.
I have to acknowledge all the department, biology department, and Ashwarya, who is not here today, but she's now Hearts for sure. And she worked my job while I was maternity leave, so I have very strong bonds to Ashwarya. And obviously all the students and all the TAs and instructors for Gen Bio for the last 4 years, because they made it totally possible for their patients and their really patients, because we had a lot of bumps on the road, but I think we're getting better and better at this. And now let's do question time. Tom, and demo. Yes, right.
So, if plants have medicinal value, as you said, do we see in some of our aspirin and other things that our shampoos, are there plants in those that we don't realize?
Thank you, Rhonda.
Not sure if that
We
makes
don't
sense.
realize that a lot of medicine actually came from plants, and aspirin is made from plants. From aspen, yes. Thank you, Dr. Willette. Willow. Yes, and so a lot of people don't realize how plants already have a crucial role in our health and well-being every day. So that's another part of this module that we can do, is just open the students' eyes to how much plants are part of our life, part of our healthy diet. So yes, medicinal properties is just one of the aspects of them, but they play a huge role in our life.
And I have another question. So that first one that you showed, I think it was the black sage, that the Native Americans used for arthritis, being that I'm getting older. Can I make that myself? If I get black, it seems somewhat easy. Black sage.
Yes, Dr. Willette would probably know all the regulations about collecting wild plants.
You just grow them yourself.
So yeah, well, I mean, so we want to be respectful where we collect our plants and stuff, but you could absolutely grow it in your garden. And yeah, so these things, we have the caveat, please don't eat anything here. And if you go visit the gardens on the second floor of LSP, there's a sign that says, please smell these things, you can feel them, do not eat them, because some of them are toxic, right. Just as Tanya described. There's a reason. They're defending themselves against herbivory. But no, of course, it's interesting in that sense. So much around us is all plant too, but then a lot of things when you're talking about the pharmaceuticals, they start that way, but they're very expensive to harvest that much biomass, and so a lot of them become synthetic. So they'll figure out the chemistry and then make it in a lab so that they can scale it up. But I do like to ask, did you brush your teeth today? Did you eat ice cream? Have you used cosmetics lately? All of that is driven by one compound, well, an agent in algae, that all of those things that touch you throughout the day, they have this compound that comes from algae that we use. And that's true throughout so much of what we walk through the grocery store, not simply the produce section, but all of the things there that are there.
Thank you.
Good questions. Yes, please.
For students who were working from home, did they receive toolkits? Maybe that was sent to them?
So the question is, students who are working from home, did they receive toolkits in our class? We have 250 students. Yeah, so as one of our students who did this online, no, we test them. But think about the challenges there. So then how do you make sure that the experiment you're running in your kitchen is the same as 3 other students who might be on the other side of the country or in another country? How do you coordinate time zones and all that? And we encountered all of that in this. And it's so like, get a cup. Well, what size cup? Is it clear? Is it ceramic? Is it plastic? Is it glass? All of those things actually, our students had these new challenges to encounter in that process. So for our students, for this undergraduate, for the freshman level course, our first year course, no, they didn't. But their upper division, it was really impressive what our other faculty did. They did send care boxes of biology boxes in the mail and things like that. But I think that was a lot of places that tried that. But because of the sheer number of students and where they were around the planet, it just wasn't feasible. Yeah.
I want to add that one of the students who is sophomore right now, Jackie, she's here. She took this lab online. You can ask her questions about what she did afterwards. She published a paper with us on the remote yogurt maker, and she was one of the students who developed her own methods to measure yogurt viscosity, because the more viscous the yogurt, the more successful fermentation process was. So they figured out the way how to create a splat test. When you drop the yogurt from the height and it splashes, and you measure the amount of splat, and based on this, you just correlate it to viscosity. And they developed another test, which was a run test, when you tilt the surface and you put the yogurt on it and just measure how quickly it runs down the surface. All of those methods were developed by students because we didn't know how to measure viscosity of yogurt at home. But students figured it out, and they found a way to how to do it as a group and as a team, and it was a really group effort. And when I was sitting in the presentations on Zoom, I was just heartwarming how much work was put into it, how much heart was put into it, and the presentation was stellar. I was like, oh my God, guys, you're the best, seriously. So that was a great learning experience for us, being online, that's for sure, even though it's off topic.
But we're very happy to be back.
Exactly.
But we know we can do it, and because we were forced, or their students know they can do it because they were forced to do it, and it was very successful to get through there. So in that sense, and so, like as Tanya mentioned, several of those things that students developed at home, we're bringing them into a lab because we're like, that's brilliant. Let's try to do that in this controlled setting.
I'm just curious about what's next. Like, does this method and do these research continue for the next 4 years, or do the results become a foundation for a new type of experiment, or what you guys envision with that?
I'll take that. Well, first of all, this is just a couple of weeks of experiments by first-year students. We definitely want to take it further. That's why 4 students this summer will be repeating those experiments into publication state, so we can start publishing the data at this point. Second, this project keep growing and keep morphing. I'm sorry, TAs, but it's going to be like that probably forever. So another student at DTE sitting here, she will be developing antifungus assays this summer. So the bottles that you saw with little balloons, they didn't really work very well because we did them just on the fly without doing preliminary testing. So what we want to do this summer is optimize this protocol so we can actually test the antifungal properties of plants next year. We haven't done it yet well. So this is one of the aspects we want to change. And we'll probably come up with some more. I really want to test anti-inflammatory properties. I don't know how yet.
Cool. And then to that point, it's not isolated in biology. So we've had multiple students who have come and said, can we dive into those compounds? And that's where we can bridge with our chemistry, and the biochemistry department is speaking with OChem. Additionally, we often vertically integrate to our upper division biology courses. There are the opportunities to take things that they're learning. And again, this is all in the scope of 5 weeks we do this entire activity, because we have 5 weeks with bacteria, 5 weeks with plants, 5 weeks with animal. So we're just scratching the surface on what's possible, but then we're really excited. What other assays can we try? There's others that suppress tumors that we just haven't figured out how to do that yet. There's an assay where we can try to suppress tumor growth. But again, so much more with that. So yeah, we're going to be playing with this for years. And maybe the next time we talk, we have 12 assays that can talk about all these things. And not every assay is going to work for every plant, and there's over 250,000 plants on our planet. So we have very good job security.
So we're not going to repeat it, that's for sure.
questions, that's great. You want to turn people loose, that's great.
Yeah, let's walk
We should
around.
definitely take a look.
Okay.
Things down here. So ideally, you would, well, I don't want everybody to start from here, but keep in mind that logically speaking, we have fresh plants here, then plant extracts and dry plants here, and then we have two stations with bioassays, which is a brine shrimp and antimicrobial and Daphne. And TAs, if you can hang around those stations just for 5 minutes. Love it.
These are all moving very slow.
Alright. So this is Artemisia.
And like what they do and stuff like that.
So can I just go get some
Yeah, just peel the leaf.
Once there's an applicant.
focus it on the ones that are moving.
At my Kelsey does not have